Here again, come here. No, the one behind you. That guy, I know he's sitting there yesterday. Come on. I want you to be sitting in front. Who was sitting in the last bench yesterday? Raise your hand. Okay, I'm going to swap people. You guys are going to go back, you come up forward. Okay. All right, I like this. You were sitting in front yesterday too, right? Okay, you want to go back there? I want you to come here. Okay, you go back there where he was. You also. Okay. Is this clear? You guys are MSCs and PhDs. Tomorrow I expect you guys to do this on your own. I don't need to tell you where to sit, okay? Is that clear? Okay. All right, we want somebody next to him. Oh, we don't. We have pairs already. Good. You guys come up here. Why are you alone? Where is your buddy? Were you there? You want to go sit next to him? Okay, you come up then. It's okay. Come up. Come back up. Okay. You guys sit there. Okay. You guys want to move there behind those two girls? Okay, I want everybody in the one line. Like you guys understand? Move there. Okay, again, this is not a 5th grader class or a 10th, 7th grader class, guys. You are PhDs and MSCs. Do this on your own, okay? Assemble properly, all right? Okay, we're going to do this again. I want everybody to tell me your, not me this time, you're going to tell the class. So you're going to move that way or, so you're going to move towards the majority of the class, okay? You're going to say, hello, good morning. My buddy's name is, right? My buddy is in which grade? And which stream? And what do they find interesting about science? Okay? What's the most exciting thing you've observed in biology? All right, let's start this side. Okay, you're gonna go on. Oh, okay, sorry. Yeah, give yourself a minute. All right.
Hello. Hello, hello. Hello. सर
Yeah.
वो वाला नहीं काम कर रहा इसको लगा देता हूँ निकाल देता हूँ
Okay. All right, I don't want you sitting next to your friend. You go there, behind her. Okay, we'll start with you later, right? Okay, you go ahead. Make sure you're sitting somewhere next to somebody who you do not know. उसको भी जानते हो तुम? Okay, you sit next to that girl. Prachi is your name, right? He's gonna sit next to you. Do you know him? No. Okay, then he's gonna sit there. You come here. You're gonna sit here. It's okay. Come on. We're gonna break the girl, boy thing here. Okay? You can sit next to the boy, girls can sit next to it. It's okay. All right? We're all professionals here. You know? All right. You're gonna do a lot of co-ed stuff in your life. All right. Go ahead. Not to me. What did I say? You're going to speak to the class. Speak loud, everybody. Calm down. Focus. Okay. Please respect your colleague when she's speaking. Okay? All right.
Good morning, everyone. My buddy's name is GIAN. He is currently pursuing bachelor's in biotechnology. His interest is in physiology of the body. Thank you.
Good job. What does physiology of the body mean? Okay. Good. You're going to tell that guy. What's your name? Sorry. You would ask me the cell question yesterday, right? Who asked me the cell question yesterday? The guy who was telling me the cells die. Huh? He's absent? Oh my God. Okay. All right. I have a pair for you for tomorrow. Okay. All right. Yeah. Please go ahead.
My buddy's name is Swati Ninja. She is pursuing
To the class. To the class. You're speaking to the class.
she is interested in first time she got interested in how flies show luminescence color.
Okay. Great. What else? Did you do all of the things? So your name is, I'm sorry again? Swati. Which what is she in?
BSc biotechnology.
BSc Biotechnology. Great. And she's interested in colors in life.
Flies.
Colors of flies.
Interested in
Awesome.
they show color.
Awesome. Great. Thank you. Sit down. Can you guys who are all from West Bengal stand up? Who's from West Bengal here? There were several people from West Bengal yesterday, right? Stand up. Okay, more? All right, sit down. People from Odisha, stand up. Okay, good. You guys are not sitting still. I'm just checking. All right, good. And people from Bilaspur, everybody stand up. Right? Okay, stand up, stand up, stand up. Who's from Bilaspur? Okay, there's only one person. Why do you have two of you are in the same bench? I don't want two people from Bilaspur sitting on the same bench tomorrow. Is that clear? Okay, good. How many of you are from, sit down please, Chhattisgarh? All right, I don't want two of you sitting from next to each other. Is that clear? Okay, so you're not going to sit next to the person who's from Chhattisgarh tomorrow. All right? Or from your city if there is not enough. All right? Okay, please sit down. Good job. All right, please go ahead.
Good morning, everyone. My friend's name is Suman. She's from Maninga, Chhattisgarh. She's currently pursuing MSc Biotechnology. And what she finds fascinating about biology are snowflakes.
Snowflakes. Okay, cool. All right, I'm learning new stuff. Go on, please. Oh, man. Okay, everybody's interested in color of butterflies. Next one. No, no, you. This way. We're going this way. Yes, ma'am. She's from? Okay, why am I hearing sounds when other people are speaking? Again, you guys are BScs, MScs, and PhDs. Guys, I don't have to control your discipline you like. Okay? When somebody's speaking, you give them respect. Right? Is that clear? Have your teachers taught you that? You too, you're not going to sit there. You're going to come up front. Okay. Where? Sit next to them together. We need two people on the same one, and we need two people who don't know each other. Okay? All right. Sorry, go ahead, please.
Good morning, everyone. She's Neena Patel, and she's from Raigad, Chhattisgarh, and she's pursuing MSc Microbiology, and she finds topics related to genetics very interesting. Thank you.
Good morning, everyone. My buddy's name is Yogisha Yadav. He is from Bilaspur, Chhattisgarh, and he finds interesting topics and innovative methods.
Go ahead. Yeah.
Good morning, everyone. My buddy's name is Sandeep Kavej. He is from Kharshia, Raigad. He is doing MSc in Microbiology. He is interested in studying proteins.
Good morning, everyone. My friend's name is Moses from Basai. He's from Rajasthan, Chhattisgarh. He's currently pursuing MSc Masters in Meteorology, and he is interested in the study of cells.
Okay. Go ahead.
Good morning, everyone. My buddy's name is Vidhi Mandavati, and she is pursuing MSc in biotechnology. She is interested in how a cell efficiently works in human's body and how a particular cell efficiently doing its work. Good morning, everyone. My buddy's name is Arpana. She's from Bihar. She's pursuing her master's in biotechnology and cell theory. Like, she has interest in how genes work, especially in brain process. Hello, everyone. Good morning. She is Shreya Gaikwad, and she's from Bilaspur, Chhattisgarh. What drove her love for science was how beautiful the science is, that it changes the caterpillar into larva and pupa and finally into beautiful butterflies. Good morning, everyone. My friend, she is Prachi Agarwal, and she's doing her PhD in cancer biology. And what she finds interesting that when she told me that when she was in class sixth or seventh, I think, we used to get that activity books of science. Yes. So in that one activity was there about vascular bundle, that how white roses used to get colored when we put it inside the colored water. So that she did, and she was very much amazed about that.
Great. Next.
Good morning, everyone. My buddy's name is Khushi Mehta. She is from Odisha and currently studying in bachelor's in biotech. Her interest is in for exploring the various life forms.
Okay.
Hello, everyone. My buddy's name is Vikas Mahodaya, and he is from Maharashtra.
Okay, we're gonna go to you. Yeah.
She is pursuing her PhD in a pharmacy department, and her interest is like how she uses field pharmacy because in 12th she has chosen a biology subject as her for 12th, and then what field she is interested is like pharmacology. There's one subject in biology, so that's why she will become a doctor.
Great. Keep going.
My buddy's name is Shivani Chaurasia. Um, She is from Chhatarpur, Madhya Pradesh, and currently she is pursuing PhD in biotechnology. And what finds me interesting in biology is when she was in her bachelor and studied about microorganisms, then develops her interest in gain more knowledge.
Great.
Good morning, everyone. My buddy's name is Om Das. He's from Ambikapur, Chhattisgarh. And his interest in science began when he noticed that a single seed develops into a huge tree.
Awesome. Next.
Another friend, she is Soumya. She is pursuing her UG fifth sem, and she is from Bilaspur only. And she was curious about how human physiology works, like the universal central dogma process is same in every creature, but how the complexity of human physiology works together. She is curious about that.
Some.
Hello, everyone. My buddy's name is Pratyusha Rajak. He's from
Pratiksha.
Pratiksha Rajak is from Patni, Madhya Pradesh. Currently, she is pursuing MSc third semester in biotechnology. And her interest in biology develops from the genetic modifications of like BT cotton.
Okay. Please go. Please keep going.
Good morning, everyone. My buddy's name is Chaya Devankar. He is pursuing a BSc Biotechnological Chemistry. She belongs to Chhattisgarh, and her interest about the human body, how the different organs of the body work in a minute or second.
Awesome.
Hello everyone, my buddy name is Balaji and he's from Hyderabad, Telangana. Currently he is pursuing MSc Biotechnology first year. And what fascinates in biology is grafting, that how grafting can produce two veggies along tomato and potato in one plant.
Morning, everyone. Today, my buddy name is Isha. She is my classmate. She is pursuing her MSc Biotechnology in GGU. And what fascinated her in her childhood is like we eat egg every day, right? अंडे से चूजा कैसे निकलता है? क्यों निकलता है? जो कि हम खाते हैं ना, अंडे से चूजा निकलता है और वो चूजे को क्यों नहीं खाते हैं? वो अंडे से चूजा क्यों निकलता है? That fascinated her.
Awesome.
Good morning, everyone. My buddy name is Divyanshu Nayak. He is from Ramghata, Odisha, and presently he is pursuing his MSc in microbiology, and he likes to study environmental health.
Good morning, everyone. My buddy's name is Das Kataria. He belongs from Akola, Maharashtra. Currently, he is pursuing PhD in neuroscience. He likes to study about brain disorders, and he has a senior research fellow in CSI.
Awesome.
Good morning, everyone. My buddy's name is Aditya Narayan. Right now, he is pursuing MSc in
You're the
biotechnology.
musician, singer guy. Aditya Narayan. Udit Narayan's son, his name is Aditya Narayan, right? Man, we gotta hear some song from you. On the last day, last hour, we're gonna do fun stuff. Okay? All right, everybody be prepared for that. I'll sing a song for you guys. I'm just
Right
kidding.
now, he is pursuing MSc in biotechnology from Guru Bashidhar Vishwavidyalaya. Basically, he is from Munger, Bihar. And one interesting thing which excites you is studying the gene editing techniques like CRISPR-Cas9 technology. He also wants to study that how antibody antigen interactions take place, and beyond that, beyond science, he is not very much serious during the lectures.
Awesome. I love your interest. Actually, on the last day, I'm going to have you guys volunteer to come up here and teach the class for 2 minutes. 2 minutes. Okay? You guys want to do that?
Yes, sir.
Okay, lovely. In the area of interest that you guys are making up. Most of you probably made it up like in last hour, right? Okay. All right, please keep going.
And with my buddy, he is 71 and he is currently pursuing his bachelor's in biotechnology at GIAN itself. And he is particularly interested in immunological science, like a disease called alopecia. He is specifically interested in that disease.
Awesome. Why alopecia?
Around 5 6 years ago when I was diagnosed with it. And at that time when I looked it up, there was no concrete like proof as to why it occurs. And recently, a funny thing happened. You asked us to read papers, so I read a paper, and turns out that there is epigenetic causatives behind it. Turns out that the HbA1c expression increases, and that somewhat leads to it.
Awesome. How many of you have heard of alopecia? Only a few. Who wants to stand up and tell me about alopecia other than Salil, of course?
Sir, moreover, in addition to Salil, in addition to the discussion of Salil, alopecia is the complete baldness, which deals with the, you can say, the complete escalation of the hairs. And after means, whenever we people talk about the treatment of alopecia, so right now only we can say we can mitigate or we can prevent. Because after a hair transplantation, lot of we can say post complications are also related to the hair transplantation.
अरे भाई मैंने तो खाली alopecia क्या होता है बोला. Good job. Keep going. Make your deficiencies that you're born with your power to do something interesting. That's what I want to tell you guys. Okay. All right. Keep going, please. Good. Go ahead. Yeah.
Good morning, everyone. My buddy's name is Dhameshwar Prasad, and he's currently pursuing his master's from Government Naval College in Dongargarh. And his interests in biology include plant science, which he is really fascinated by the medicinal uses of plants. And he's also interested in proteomics, which is the study of proteins and amino acid sequences.
Awesome. Do you guys know many of the medicines come from plants, right? Good. Which is amazing, actually, to think of it. All right. Keep going.
She's interested in cancer biology and clinical microbiology.
तुम लोग को 2 मिनट बस 4 लाइन याद करने बोला है, राइट? Awesome. You guys want to come up forward and sit, please? Okay. Don't leave a seat behind. Okay. Same thing with everybody. I don't want a seat empty. Okay.
Good morning, everyone. My buddy's name is Ratna Dubey. She is from Bemetara, Chhattisgarh, and she is a PhD scholar in botany department. The topic that interests her is stress physiology. It fascinates her a lot that how these physiological characters, the plants, they adapt to different challenging conditions.
Awesome. You know what fascinates me about plants? Have you guys seen this बेल का tree, right? बेल नहीं, क्या बोलते हैं इसको, जो जाके पकड़ता है. Like, have you guys seen that? That's amazing. To me, every single time I see it, it's like, wow, the biology is so brilliant, right? It can go on and just figure out where the support system is, right? That's bale, right? What is it called? It's climbers, right? Somebody has to tell me the science behind that someday. The plant guys. Okay.
Their interested topic is microbiology and currently pursuing PhD from botany department.
But what is her interest? Did she say? Huh?
Microbiology.
नहीं, life में क्या interest है? I'm not asking that. I asked about what is it that excites you about science, which one phenomena that you find exciting. You guys didn't discuss that? Go ahead.
So, when I opted for microbiology as my bachelor's, the first thing that came to my mind that how these tiny little organisms can lead to so many things. Like they can be harmful, they can be useful as well. So, this one thing excites me towards that.
Okay, great. You know, in our body, how many microbes live? Huh? How many? Billions of microbes. How many human cells are in a human body? Somebody with a, no, I said DNA, that was DNA basis. How many human cells are in your body? How many trillions? Somebody stand up. Nobody knows this? About 37 or so trillion, 35 to 37 trillion cells of human body. You know how many microbes live in your body? 39 to 40 trillion microbes in your body. And most of them live in your gut. I actually have a startup in Bangalore that is on microbiome, gut microbiome. And I start every presentation to an investor with this particular slide. So we have more microbes in our body than human cells, just by numbers. You know how many genes those microbes code for? About 2 million genes. And you know how many cell genes we have in humans? Where is it? How many genes do we have in humans? We discussed this yesterday. About 23,000 protein coding genes. How many genes the microbes in your body have? 2 million. Can you believe it? You cannot live without the microbes in your body. Do you guys know that if a mouse that is born and kept in a microbe-free environment, it's called germ-free environment, they do not survive past a certain age. What does that mean? It means that you are because of your microbes. Okay? Isn't that amazing? I find that amazing. Majority of your health on a normal daily basis is contributed because of microbes. And we eat lots of antibiotics for no reason. Okay? Anyways, please go ahead. Who was the next? This is an epigenetics class, not a microbiology class. Who was next? You guys. Go ahead. Yeah.
Morning, sir. My buddy name is and she is pursuing MSc Biotech, and she is interested in food and industrial microbiology.
That's a boring answer. I need to understand what phenomena in biology interests you. Okay? Okay, cool. Keep going. And we want to, sorry, end it a little quick. So just 20 seconds. Okay, everybody, 20 seconds.
How they are related to many food industries and
Great. Go ahead. You guys sit there. I don't want you guys to be sitting behind. If there are empty things, you're going to sit there. Same thing with you two. Is that clear? Right? I don't want people sitting behind a certain row. If things fill it up. Okay.
Good morning, everyone. My friend's name is Shivani Singh. She is from Dantewada, Chhattisgarh, and right now she is pursuing MSc Biotech Culture. And she finds fascinating about bioinformatics and computer-aided drug design. That how the computer is very useful in designing the drugs, and it does not involve any volunteers all the time.
Awesome.
Good morning, everyone. My buddy name is Jyotika. She is from Odisha, and she is doing PhD in microbiology.
What's her interest?
We don't know each other much, so
That was the interaction for, no?
that was a bit late.
Class starts at 10:15. Okay? Class starts to everyone who came late at 10:15. 10:15 means 10:15, not 10:17, not 10:30. Okay? 10:15. Okay. Okay.
My buddy's name is Vikas Nande. He's from here only from Jasper, and he's pursuing third sem in MSc from here only. Uh, his uh, his interest is like the modifications in DNA and how epigenomics is related to that.
Awesome. Somebody likes epigenetics. Okay. Keep going. Next. Yeah.
Good morning, everyone. My buddy name is G. Sreyans. He is a student of Histone, in biotechnology. Her mother is a nurse, so he has interest in nursing, like pediatric and biochemistry.
Awesome. Do you guys know what to do? Yeah, your turn. No, behind you. See. Do you know what we are doing? You came late. Right? Do you guys know what we're doing? No. Who does not know what we're doing? Okay, we'll do this in next class with you guys. Okay? All right. But please come on time. I am the only one who's allowed to come late. Okay? Sorry. All right. We're going to start here. Okay, you guys just come on.
Prachi, and he's from Bilal, Chhattisgarh. He's doing a PhD in gynecology, and he eventually developed an interest in biology from microbiology, and when she experiment on the gram-string bacteria and all, then he feels that it is exciting work, and then she develops.
Go ahead.
His name is Dr. Mukherjee. He's from Hooghly, West Bengal. He develops his interest in biology by reading biochemistry, basically, by reading about how these metabolites regulate or controls our body and all. And he is also interested in reading molecular biology, gene expression.
Awesome. How many of you came to this field because you had no other option? Good. I like honesty. Good. All right. What are we going to do tomorrow for introductions? What do you guys want to do? I know it takes around 30 minutes, okay? But it is important for you guys to be able to come up and speak at least. Some of you don't even do that well, right? Learn to speak well with confidence in front of everyone. That's what I'm trying to teach you guys, okay? And know yourself. Okay, I mean, my job is to teach you epigenetics, but I understand that personality is equally important. So get up, speak, be able to talk to unknown people, be able to comfortably sit next to them, be respectful of the people. This is important, right? This is important traits. Whatever you do in your life, you're going to have to do that every day, right? Okay, cool. What do you guys want to do tomorrow? Hypothetical startup ideas. That's going to take at least 2 hours. I would love that, but if you guys can finish it in 5 seconds, sure. Any other ideas? What would you like to know about your friends? It's not about me. I'm not going to remember any of this. I'm sorry, I'm old. But you guys, it's about you guys knowing each other in this class. It's not about me. It's about you guys knowing each other, okay? I will probably remember like 3 names and maybe 2 of you where you came from. But I'm sorry about that. But that's not, I mean, the intent is for you guys to know each other. What would you like to know about your friends here in the class? You don't care about your friends, right? No? Anything? Yeah.
Where my friend wants to see herself in next 5 years.
Interesting. Let's do that. Okay. I want to see tomorrow you guys figure it out. Tonight, only one day to figure out what you want to do in the next 5 years. Okay? All right? Okay, good. I have this. I wanted to start here. A simple thing like what we were discussing yesterday, right? What's on the screen? Somebody stand up and tell me. Guys, come on. You can speak up. What is it? Yeah, go ahead. Awesome. My name is? My name is not Sir. Okay? My name is Kunal. I want you guys by the end of this course be calling me Kunal, not Sir. Okay? I'm giving you 5 days to practice. Okay, good job. I want you guys to appreciate, we discussed this yesterday, that our genomes have majority of the portion of our genome is. Do you guys know what genome is? What is genome? 6.6 billion of bases. That's genome. Okay. Um, okay. Before we go anywhere, actually, I want you guys to see my laptop, unfortunately. So, come on up. Come on up here. Everybody, line up. You guys see my beautiful laptop? Right? Can you see this? What is this? Nothing. This is a software called IGV. Okay? Integrated Genome Viewer. It's from Broad Institute. Okay? It's one of the most frequently used softwares. I'm not propagating Broad Institute, but this is the most frequently used software which you can easily use. You can go download it for free. Okay? It requires Java on your computer, but okay, here, right? So I opened up something. I'm gonna open here. I say all chromosomes. Okay, on the simple screen, do you guys see here? There are chromosomes listed, right? Okay, this means nothing right now to you, does it? No. Okay, I expect that. How many chromosomes are there? Forget about all of these. Let's go to chromosome 22, X and Y. We have 23 chromosomes, 22 autosomes, 1 X, 1 Y, right? Do you guys know of any gene names? Somebody tell me a gene name. Eh? Huntington gene. What's the abbreviation for Huntington gene? I don't know. Anybody else? Anything else? HTT. Okay, we're going to see HTT. Can we see HTT here? Oh, we can. Awesome. Yeah, I'll show you guys. Don't worry. 1 second.
Sir, what's the name of software?
IGV. Here's a gene called HTT, Huntington gene, that causes Huntington disease, right? There's a gene here. There is a gene next to it, which is called HTTAS, antisense, okay, which essentially controls Huntington gene's expression. It's a non-coding RNA. We'll talk about these things, okay? Why am I showing you any of this? Do you see this? Okay, I want you to see, appreciate this. There is one gene, there is one transcript variant, there's another transcript variant. There's the gene next to it which has tens of transcript variants, okay? And I couldn't show you this on the screen, of course, because I don't have IGV on that computer. I can't hook up my computer there, okay? I can zoom out. I'm zooming out. I can see genes on this chromosome. Can you see? There are many more genes that are coming through, right? Now you go, keep going. There is spaces between the genes. This is non-coding. There's a gene, small gene here, small gene here. There's a large gene here, which is HTT. You guys see the HTT again, right? There are other genes here. You see all the genes? NSD2, this gene here has many variants. Okay, transcript variants. What does a transcript variant mean?
Various form of mRNA.
Various form of mRNA that gene will make. First lecture right now is going to be all about transcription, and I want you to appreciate what a gene means. Okay, okay. We're going to zoom out here again, and you see lots of genes on this chromosome. This is chromosome 4. Sorry, I'm blind. Chromosome 4. You see this? Okay. So all the genes you're seeing now on chromosome 4. What's up? Did I say I'm blind and that's funny? Okay. Here is a centromere of the chromosome. Do you guys understand this centromere? There's a centromere here. Centromere here. There's 2 beautiful red spots. Okay. Here is the end of the chromosome. Those are called telomeres. Very good. You guys know your stuff. Then we'll be done today pretty quickly. Okay. I want you to go into centromere. Okay. Centromeres. Let's go to chromosome. Okay. Because you guys see the gene structure? I just want you to appreciate the gene structure. That's all. Okay. Gene structure on a chromosome, non-coding DNA here. This is all non-coding. It doesn't code for anything. There are lots of genes here which are called AS, which is antisense. I just told you that on a HTT. You can keep moving. You can keep observing the genome. Right here. Okay. All right. See that? Okay. I'm going to go to, okay, that's all I want to show you. No. Let's go back to HTT. Just because it's favorite gene of somebody who comes from Angola, right? Okay, you guys see this? There is exons, which are the boxes, and there is the rest of it is called introns, right? Okay. Okay, where am I? The gene goes, where is the arrow? Arrow is pointing to this direction. That means it's going from 5' to 3'. You guys know all of this, right? Wow, that's cool. That means I'm not in the bad class. Okay, and this is where the gene starts. You see this region? That's your 5'. This is your transcription start site right here. This is your 5' UTR, right? Okay, good. All right, go back to your classes. Go back to your. I want to show you this. You guys can go and play with this if you're interested. But what's beautiful about this is you have a chromosome. There are lots and lots of genes around this. There is non-coding RNA that is in the intervening region of the genes. And a gene has a structure, right? Is that clear? You guys know this stuff? Very cool. Did I teach you something new there? No? That was all stuff you knew, right? Okay, we started this yesterday. There is 3.3 times 2 billion bases, or 23,000 genes, 2% of the DNA. More than 97% of our DNA is non-coding. That's a paper. It's a Nature paper in 2001. Classic paper I told you guys about yesterday. That's first human genome sequence. Okay, that's where we figured out, oh my God, we thought it was all genes. We've got lots of regions that is we did not appreciate the extent of non-coding RNA, non-coding DNA that is in our body. You guys were just born then, most of you, right? Or we are being born. Okay, all right. And that's what we just looked at, right? A gene structure. Right? Do you guys know all of this? Okay, so we've got an enhancer. What is an enhancer? Okay, we'll talk a lot about that later on in the second part of our thing. Okay, enhancers are key. This is a gene, there is an exon, there is an intron. We talked about 5' UTR. We talked about start codon, right? Start codon is not the place where gene starts. That's called transcription start site. The start codon is where the translation starts. Okay? Stop codon, start codon, stop codon where protein ends. I'm sorry, I'm going to have to go back up. Oh man, this does not work. Okay, go back. Stop codon, 3' UTR. UTR means? Great. You guys know your stuff, which is, I'm so happy. Polyadenylation sequence. You know, polyadenylation sequences, poly A at the end of the mRNA. That's going to make your RNA happy. Do all genes have poly A sequence? Do all RNAs have poly A in them? Which RNAs do not have a poly A sequence? Okay. Which RNA do not have a poly A sequence? Have you heard of ribosomal RNAs? Huh? Okay, all right. Ribosomal RNAs don't have poly A. mRNAs, not every single mRNA has poly A, by the way. Okay. It's promoter, there is cat box, there is Tata box. We'll talk all about this, GC box, whatever that means. But I want you to appreciate a structure of a gene. That's what I was trying to show you on IGV viewer. You can see every gene is different, right? All right, you guys know this. We talked about this yesterday. What is this? Central dogma, right? This is transcription, replication, reverse transcription, translation, makes protein. Beautiful. Everybody knows this? Who does not know this? I do not like the word central dogma, but all right. Okay. We're going to talk about transcription in the first hour. Okay. Why do we talk about transcription in an epigenetics class? Why do you want to devote an hour to transcription? Come on, yesterday we talked about all of this, guys. Why do I care about transcription? As an epigeneticist, why do I care about transcription? What? I'm sorry, I can't hear you. Can you speak loudly? mRNA does not code for a protein. That's why I want to know about transcription. That's okay. Okay, I see a lot of people coming in. Are you guys enrolled in this class? Or you don't want to, you're just coming to have fun? All of you who just came, are you guys in this room or you're just coming to learn? You guys are in the university, right? Why are we sitting in the back? So that we can look at our phone, YouTube videos. Okay, come on up front. If you guys want to hear me, I can't hear you from there. So please come front. Come in front. Come in front. I don't want anybody sitting on the back bench. There's lots of room up. Come on. Yeah. We care about please. Thank you. We care about transcription because without transcription, DNA cannot become RNA. Of course, that's what transcription means. Why do I, as an epigenetics person, care about transcription? My meaning of whole yesterday's class will be zero, guys, if you can't answer this question.
Epigenetic variations make various transcriptions.
Okay. Okay, what else? Up front, I want you sitting on the back, all you guys on the back. Are you here for learning or are you here for something else? Good, that's a good way of saying it, but I don't care about DNA methylation. Why as an epigenetics person should I care about transcription? Again, I need a better answer. You guys are getting there, but you have your... What did we learn yesterday? What was the message? What was the take-home message yesterday? Understanding transcription is crucial to understand expression of genes. That's right. Why do I care about transcription as an epigeneticist? What was the take-home message yesterday? Proteins don't come into picture at all. We're just talking about transcription. I do not care about protein. As a person, as an epigeneticist, I don't care about protein. I'm sorry, that's not true. But I mean, for you guys, I don't want you to care about protein. Guys in the back, please come up. If there are seats up, please come up. Okay? Can you hear me there? I cannot hear you there from there. I'm deaf. I can't hear. Okay? All right. What was the message yesterday? I'm sorry. I'm just going to ask again.
Some modifications in the transcription process can reverse the pre-genetic alterations.
Okay. Transcription जरूरी होता है, okay? But as an epigeneticist, why do I care about transcription? Okay, what was the take-home message yesterday? I'm just asking two questions again and again. What was the message yesterday?
Transcription is the first step that will lead to the phenotype of the trans.
Okay, but why do I care about transcription as an epigeneticist?
Because it is important for normal physiology.
What is important for normal physiology? Transcription?
After the transcription, we identify the coding and non-coding.
Okay. All right. Maybe I'm just not hearing you guys out. I care about transcription. Yeah. As an epigeneticist, why do I care about? You're getting there? You're getting there? As an epigeneticist, why do I care about transcriptions? कल क्या पढ़ा था हमने? Different phenotypes, exactly. How do different phenotypes occur?
Because splicing can take place in the transcription, that's why the functional genes can produce the protein.
Okay, I gave an example of aldehyde dehydrogenase and neurotransmitter. What did I say? Exactly, exactly, exactly. So why do I care about transcription? Okay, I care about transcription because I want aldehyde dehydrogenase to be expressed only in the liver cell. And epigenetics controls that. I told you this yesterday, right? Right? So what do I need to understand? I need to understand how does my epigenetic modification leads to specific expression of aldehyde dehydrogenase only in the liver and not in the neuron, right? So I care about transcription. I care about transcription steps to be able to know how does an epigenetic modification control expression of a gene. Is that clear? Right? Okay. Why, as an epigeneticist, why do I care about transcription? Who wants to tell me what I just said in your own words?
It leads to specific transcriptomics in different states.
Yes. So I care about a transcriptional pattern that occurs only in the liver and not in the neuron. Some that happens only in the neuron but not in the liver, and vice versa, and so on and so forth in different organs, right? So I care about how epigenetics controls transcription at different level, right? That's what I'm trying to do. So I need to understand what transcription even means to begin with, right? What are the steps of transcription so that I know where does epigenetics come into play? How does epigenetic control transcription, right? That's where my epigenetics comes into play in the daily life on an every single cell, right? Is that clear? Or did I speak in Marathi? Is that cool? Everybody understands English? Okay. In the next class, I'm going to ask you the same question again. And I will appreciate an answer. What does this slide say? We know this stuff, right? What is on the screen? I have a DNA. Everybody recognizes DNA, otherwise you can get out of the class. And then I have a big protein sitting on that. What is that? I have an RNA polymerase. Everybody knows that. What does a polymerase even mean? Okay. All right. When I answer a question, one of you should just stand up and answer the question. And that person who stood up and answered the question should not answer the second question that I ask after that. Okay. But I want you guys to just stand up and tell me the answer that you know. You express yourself. It's okay. All right. Express yourself. Okay, there's an RNA polymerase that's making a beautiful red RNA, right? That's coming out, and that's going to make our beautiful protein that we desire to make. My protein's name could be aldehyde dehydrogenase. Its name could be neurotransmitter X. Its name could be anything, right? That we wish. Huntington, for if you will, right? Okay, all right. Basic steps for transcription. This is undergrad stuff, guys. This is really not, your level. You guys are smarter than this, right? You guys all know this, should all know this. Do you know this, right? Okay, what is on the screen? Oh, I just said one person stand up and answer. Didn't I? This is basic steps of transcription. This is from a book called Molecular Biology of Cell from Lodish, Baltimore. If you guys have heard of it and read it, I'm sure you have. If you haven't, go on, read it. Okay, let's go back. Initiation, elongation, termination, right? I'm bookish stuff, guys. You guys are really good at this. I'm not going to go on and spend my time with bookish stuff. I just want you to appreciate transcription is a complex process. It requires 3 basic steps. Initiation, as it says, initiate transcription. Elongation means you initiate and then you elongate, meaning you're ready. Okay, I got this. I pull my trigger, and I'm going to go all the way to make my gene transcribe, right? That's my elongation. At the end of it, when I terminate, I have to terminate. And what does termination involve? Polyadenylation, etc., etc. And I'm not going to care about that today, right? Because that's not where epigenetic regulation plays a huge role. Epigenetics regulation plays a huge role during initiation, transcription, initiation, and elongation. That's where a lot of that happens. In fact, it might play a role in during termination. We just don't understand it. So there's a lot of research in this area. There's lots of beautiful people doing amazing stuff. This, whatever you have on the screen, is fundamental to you. This makes you who you are on a daily basis. This happens in seconds in your body. Okay, this is beautiful stuff. The cell is amazing. It knows it's better than an engineer. It can do all of this stuff within. Okay, all right. So I said. Initiation is the process where lots of regulation happens. I'm going to use the word regulation, right? What does regulation mean? Regulation means control, right? Thank you. I would love it if people stand up like that and give an answer. Okay? Next time it's somebody else's turn. Okay? What am I showing you on the screen? I'm showing you very simple stuff, guys. Very simple, okay? I'm going to keep it very simple, to the ground, so you can understand and there's no confusion. Tomorrow you don't tell me what is genome and gene and all. The third person comes says, Kunal Rai taught epigenetics and you don't know epigenetics. I don't want that, okay? Okay? Is that cool? If something you don't get, ask me. But what is this? It's a transcription. Okay, all right, let's see. Somebody stand up and answer. I'm not going into this myself. I can, but I want you guys to tell me. You understand this? Yeah.
So the following slide explains the initiation of transcription. There's a data box binding protein which is known as TBP, and then there are transcription factors which are known as TF2, TF2. There are multiple transcription factors. So the promoter region is identified by the factors, and then the RNA polymerase protein attaches to the strand.
Salil knows more than what is shown on the slide. Great, thank you. But that means I'm showing you very simply there are two things: transcription factors and RNA polymerase. Transcription factors is a big word, okay, because it means a lot of things. He said TF2P to REDUD. We'll come to all of that in a second, but I want you to simply appreciate there are sets of transcription factors. There is an RNA polymerase. They work together to make initiation happen. They sit down on certain places, which is called promoter. I showed you promoter and enhancer earlier. What is a promoter? Somebody stand up and answer. Specific sites on a gene, regulatory region, great, regulatory region from regulation, where transcription factors will sit down and start the process of initiation. Thank you. And that will recruit RNA polymerase and transcription begins. That's what the slide shows, very simply. Okay? Okay, so a gene is transcribed several times, in fact, lots of times. You know? How many mRNA copies a gene will make? Somebody stand up and tell me your answer. Different genes will make, right? Some genes that are maintenance genes, such as beta-actin, that maintain your cell, okay, will make thousands and thousands of copies. There are other genes that are regulatory in nature, they'll make smaller number of copies. Most of the transcription factors, the real transcription factors, are actually not expressed very high. They're expressed in few copies, okay? But they know how to make protein after that, okay? So a gene can make 10,000 copies of mRNA or 100,000 or can make 5. But that regulation of how many copies of mRNA will be made depends on how activated that gene is. And that activation is controlled by epigenetics. That's really why we are here, okay? Got it? Okay, so that makes some mRNA, it has to go on. So we're going to talk about steps of eukaryotic transcription, initiation, and elongation, termination. We're not going to talk about elongation, termination too much. I will talk about elongation, not termination. We talk about factors regulating this and chromatin structure, and then techniques to study eukaryotic transcription. We'll talk about that in the second hour or so, okay? But I want you to just get the process of transcription first. Transcription initiation. This is again a slide from the same picture from the same book. Okay, the next picture on that book is this. It says a eukaryotic promoter. It says a Tata box. Tata box is a Tata box. Not every gene contains a Tata box, but you can presume for a sense of it that it does. Tata box because it's a TA rich sequence. Very simply, Tata box will recruit Tata binding protein. For the lack of better word, Tata binding protein, that is called a type of transcription factor. There are lots of regions like that, which are specific DNA sequences, okay, where Tata protein will bind. There is a start point, is the transcription start site. Several transcription factors bind, post binding of Tata binding protein, and then transcription initiation occurs, and RNA polymerase is recruited, and that goes on, okay. Not very simply, transcription factors come, polymerase goes, transcription begins, simplistically. You get that? That's you got transcription factors. What is transcription factor? I've been saying transcription. Transcription factors are proteins involved in the process of transcription that initiate and regulate gene expression. Why do I care about transcription factor as an epigeneticist? Why?
Because some transcription factors are involved in the modification of the
Transcription factors are fundamental to gene transcription. They actually do regulate all the epigenetic proteins that will come and regulate the gene transcription. Okay? And epigenetics is about, a lot of it is about gene regulation, right? That's what we're studying. How many of you got that? You got that, guys? How many of you are going to stand this up? Very simple things, right? A 10th grader should be able to understand what I'm teaching right now. Here is a crystal structure. You guys understand what does crystal structure even mean? No? We'll talk about that in a second. Here is structure of DNA. Here is a crystal structure of a transcription factor. This has specific motif here. You see this? Helix to helix. That's going to bind to the DNA. It has a pocket that, boom, right? Very simply. Biology knows this. Very well. Fitting one thing into another. That's what this does. You know, helix turn helix loop, it says, right? Just for the sake of complexity for you. But you understand helix. You've heard yeah. You understand loops. You understand beta sheets. Oh, cool. I don't have to do much work then. Cool. All right. Every transcription factor actually has a binding site. Okay. All right. Transcription factors are, she gave the answer, recruit regulatory proteins that are involved in the process of that initiate and regulate the ice, right? Now you got that. That's picture very simply cartoonized for you by Miss Dr. Arpan, and that recruits polymerase that starts the gene transcription. Whenever you see that sign arrow, that means gene is transcribed. If you have not got this earlier, just know that whenever it shows that, that means the gene is transcribed. Instead of an arrow like this, if it shows a solid line, it means gene is not transcribed. Okay, that's a repressed gene. Activated gene, repressed gene. Activated gene has an arrow, repressed gene has a, okay. All right, have we got that right, everybody? Okay, cool. That's now getting the complex stuff. This is RNA polymerase II structure. Okay, that's human. This is S. pombase. S. pombase, not human. S. pombase. Who knows what is S. pombe even mean? It's a yeast. Okay, all right. This structure is similar in humans. I didn't get a better picture, but nonetheless, that's the top view. That's a front view. That's a back view. This is there's 12 subunits on the C on the bottom. There's 12 subunits to RNA polymerase. I don't expect you to remember this. You don't need to care about that. Okay, just telling you how it looks in real life. That's the real life RNA polymerase, guys. Put your hands together for RNA polymerase that got Nobel Prize. I'm not kidding. Okay, great. All right. Am I entertaining enough? Right? I practiced that. Okay. All right. So RNA polymerase is the key to gene transcription. Of course, it's the enzyme, right? That's the key. RNA polymerase has this beautiful nose coming out that you see that has multiple repeats of the sequence that is on your screen. That sequence has serines on it. S for serine, right? You guys know serine. You guys know amino acids. How many of you don't know amino acids? It's okay, you can tell me. How many of you know difference between amino acid and bases? Is it clear in your head? What's a base? What's an amino acid? Amino acid makes protein, bases make DNA, guys. I made this mistake in front of my PhD advisor one time, by the way. And he was looking at me like, what are you talking to now? I was like, sorry. Okay. But this has repeats. S is the serine. Serine is very good at getting phosphorylated. It loves to be phosphorylated. And we use that for so many regulatory things in our body. In fact, your gene, your cancer, your every single thing you're made of is because of beautiful serine getting phosphorylated by enzymes called kinases. Kinases are the enzymes that phosphorylate stuff and regulate so many different things in your body. Amazing stuff. Kinases are the most drugged things. Most of the medicines you eat, actually many of them are kinase inhibitors. Okay, so this whole thing is regulated by kinases. In this case, they're called CDKs. I'll talk about that in a second. But I want you guys to simply appreciate the beautiful cartoons. As I said again and again, Tata box code promoter. We get that. Salil's talked about TF2D. This is Mr. TF2D, okay? TF2D is the first thing that gets recruited. That's a transcription factor 2D for a lack of better name, okay? You guys will have slides, so you don't have to take pictures. You will have this slide, okay? And you can go to YouTube, search animated biology, it's all on there, and you can hear better version of me speaking, okay? That's not me. But you can hear that. TF2D then recruits TADA binding protein that recruits TAF, and I don't expect you to remember the sequence of stuff that happens. I just want you to, I'm just saying it because I have to, but you don't need to remember this. It tilts the DNA, it brings something called other regulatory elements together. So gene promoter, we'll talk about that in the second half. And there you go, that's the structure. You see the structure on top? Where is the beta sheet? You guys said you know beta sheets. Somebody, come on, get up. Where's the beta sheet? Somebody said you know beta sheets. Come on. Where's an alpha helix? Nobody? Alpha helix? Good job. That's alpha helix, that's beta sheets. If I don't keep you engaged, you're going to be sleeping, and I don't care about you sleeping. I want you to be awake, right? Okay, that tilts the structure. You see the DNA structure being tilted, right? That's beautiful. The nature does this really, really quickly every single time within milliseconds, right? It's so cool. There's a protein that tilts the DNA, and the DNA will bring things together. There are a series of transcription factors that will be recruited, and the transcription will start. What's on the bottom is something called X-ray crystallography. So that's X-ray diffraction that is used to identify structure of proteins such as the one you see on top. Okay, that is called X-ray crystallography. So many of the Indian scientists are actually very good at X-ray crystallography. It's a lot of protein structure was solved here in IISc, etc., etc. Some really amazing scientists were there, and Indians somehow tend to be really good biophysicists for whatever reason, because they tend to remember the bookish stuff really well. Sorry, but yes, I'm just appreciating the fact that a lot of people actually have done this very well. All right, that's that. TF2B, 2A comes, TF2F comes. Again, I don't want you to expect to remember this. I'm just going through this. Don't worry about this, okay? RNA polymerase chain. Now, this is the crucial process. I just told you guys, right? Phosphorylation, phosphorylation, phosphorylation. If you don't remember anything else, remember RNA polymerase phosphorylation. It's a regulatory step, okay, that drives gene transcription initiation, that drives gene transcription elongation, that drives gene transcription termination as well. So RNA polymerase is the most beautiful thing that makes your RNA all the time. Once that's phosphorylated, boom, that knows how to go on the DNA, right? That's RNA polymerase starting the gene transcription. So summary so far. Can somebody want to tell me? The TBP subunit of Tata TF2D binds to Tata box promoter, distorts the DNA almost 90 degrees, that recruits the rest of transcription factors, 2B, 2A, then 2F, etc., etc. RNA polymerase 2 is recruited, RNA polymerase TF2HE and are recruited, 2H phosphorylates the CTD, which gives the transcription starting complex. This whole complex on that top is called pre-initiation complex. Okay, pre-initiation complex. Pre-initiation complex is a beautiful thing because now it recruits epigenetic proteins, where we come into picture now. We talk about epigenetics after that. Okay, so I want you to appreciate that there is this beautiful complex called pre-initiation complex that recruits proteins that will actually go on and do a lot of things that will eventually lead to gene transcription. RNA polymerase 2 leaves, starts moving. That was the summary. That was gene transcription initiation. Lots and lots of years and years of work was what just put into what I showed you, okay, by many scientists to actually understand what gene transcription even happens. Elongation, my good friend called Debabrata Biswas in IICB, Indian Institute of Chemical Biology, was my roommate actually in PhD. He works on elongation, and he's done some of this beautiful work here. And I'm not showing you his work, but I'm telling you that elongation is something that people still study today and get really, they experimentally test this every single day as we speak. Okay, so what does that mean? That means we do not understand all of this still. Okay, whatever I'm showing you is still things that we've learned so far. There are models, and you guys can become scientists tomorrow and change the next level of images people will make. Okay, that's the motivation you should have. Your name will be in the books. People will remember what's your favorite name here, right? That X, Y, and Z went on and did this work. Just like I'm taking Debu's name. He's an amazing scientist, works like 17 hours a day, literally in the lab. Okay. And I mean, people, really smart people, really hardworking people make it happen as to what you're learning, and you can sleep into the class. But there are lots of people out there who are doing this work. Okay, so be motivated, do science, understand biology, and your names will be in the books if that is a motivation, or you will just have satisfaction of finding something that nobody else has found. Isn't that cool? The cool thing about finding something that nobody else has found in life over watching a YouTube video is amazing. Okay. Okay, All right, in elongation then, what happens is there are these two factors that are shown, NEF and this other one called DSF. They work together to pause transcription at some point, okay? And they go on and they recruit something called PTFB. PTFB is the central regulator which allows the elongation to go on and happen. PTFB again now recruits the next set of epigenetic modifiers to modify and pause at this level. Okay, so transcription stopped at that place until PTFB was recruited. Okay, PTFB was not recruited, the gene was not transcribing. It just started and it paused. Okay, why did the gene build these? Why did the cell build these systems, you think? So that all those aldehyde dehydrogenases and neurotransmitters do not express where you should not be expressed. So the cells have made these complicated systems so that we can control every gene at a level where we want to control. That's why epigenetics comes into play. They bring epigenetic factors and then they go on and make things. Okay, that's what we are studying. So today will be all about this. Okay, did you guys get it, or is it too much knowledge? Get this? Clear? So initiation happens, pre-initiation complex comes, RNA polymerase goes, then pauses after some time, and you need the next set of signal for it to go on and make into a productive elongation. Okay, until elongation happens, mRNA will not be made, the gene protein will not be made, right? So we had one layer of information, one layer of regulation at the initiation level, then we have another layer of regulation at the elongation level. Okay, all right, PTFB comes, kicks the phosphorylase at NEF2, NEF, as well as more phosphorylation in the polymerase, tail that repeats. Remember 52 repeats I told you? More repeats get more phosphorylated. RNA polymerase knows now I'm going to go next. This causes, can you guys read, right? I don't have to speak this. You can read. Huh? Everybody can read the slides? Okay, please read, focus. Okay, I'm not going to say every single word that is on the slide. Okay, so PTFB can phosphorylate, that negative association it blocks, and then that causes into productive elongation. Okay, polymerase can now move along the gene body, right? We saw gene body on the first slide, second slide I was showing you. Boom, gene body goes all the way, and then can transcribe. So there is a more complicated stuff for you. Not really complicated. So at the initiation level, this kinases that I was talking about, there's a kinase called CDK7. Do you see that CDK7? That's the kinase, cyclin dependent kinase 7. Okay, the next is cyclin dependent kinase 9. Okay, so on and so forth, CDK12. So these are called transcriptional kinases. Okay, they come, they bind, they phosphorylate RNA polymerase and the rest of the stuff here and there. I don't have to go into depth of it. You just remember that there's phosphorylation events that happen on RNA polymerase that will initiate and make the RNA polymerase go. There is a different set of serines that got phosphorylated. At this, there is a different serine. At this, there is a different serine threonine, and so on and so forth. Threonines are the other amino acids that get phosphorylated. And then the third one is tyrosine. 3 major amino acids that get phosphorylated: serines, threonine, tyrosine, and all of that your body does is, a lot of it actually is due to the regulation by these 3 amino acids, phosphorylation. Okay, so there are tons of kinases in our body. There are over 2,200 transcription factors in your body, if you know. Kinases, there are over like 600-700 kinases. Okay, they play very important roles. Lots of people study one of them. They spend all of their scientists, all of their life studying one kinase, and they still can't get the whole thing done. Some people study many kinases. The CDK7s and 12s and 9s that I just showed you, people are making inhibitors to those for cancer production. There's a lot of clinical trials that are going on that actually will now lead to the next step, right? If that shows activity, then people get approved that cancer patient will eat that and they will be good, right? That is real story. I'm not kidding, right? This is what happens on research. You guys know this? You don't know this? Okay, CDK7s and CDK9 are major transcription factors in transcriptional kinases that I just told you about. People have made inhibitors to those. There are clinical trials going on to see if we can treat cancer patients with that. Okay, so hopefully somebody else will teach you that at some point. Obviously, I do cancer epigenetics, so I know all about it, and I would have loved to teach you that, but I'm not teaching you that. Somebody else will. Okay. I actually take majority of my classes that I take is in cancer epigenetics, not in this basic transcription. So that's why these slides are not necessarily mine slides. Okay, some of these slides are from Debu. So what slide you're seeing is from Debu, the guy I just told you. He's a Wellcome Trust recipient for 10 years. He is a senior Wellcome recipient. He, again, because he works hard, right? You guys need to work hard. How many hours did I say he works hard? When he was my roommate and PhD student, he used to go in lab at 9, and our deal was that who comes later. So we used to come 12 in the night or later after that. So we'd spend all that time in the lab, working 15 hours in a row, in a stretch. Of course, we'll eat in between. We can't function without eating. But what does that mean? There are lots and lots of people out there who dedicate their time to research, okay? And we're trying to solve diseases, and that's why I'm so upset about missiles and all, because we all work hard to solve diseases because we care about people, right? So if the CDK inhibitor that is being tried in clinic, there were not like 10,000 people working behind it, it won't be tried in clinic. Somebody has to go on, figure out the mechanism, which you just read. Somebody has to go on, make an inhibitor. A chemist has to go on, make an inhibitor. Somebody has to go on, figure out how's the best manufacturing for that, so that I can do a trial. A clinician has to go on and do the clinical trial in patients. Patients have to volunteer to go on the clinical trial, right? That's how drugs get made. The ones you eat that you say, oh, the doctors give these drugs all the time and I don't want to do this, and that doctor who makes money out of you eventually, right? Because you think the doctor is making money out of me by giving me medicine that doesn't matter, okay? But that's not true. There are lots of people behind that, right? Okay, that COVID vaccine that you guys have eaten, a lot of you, right, have taken. You feel like that was well, there's lots of thing out there, right? Why COVID vaccine, this does that, how many thousands and thousands of people and billions of dollars would have gone into making that vaccine, and people just say, right, why? I'm just telling you the work that goes on, right? And I'm not kidding. These things take a lot of time to really figure out what happens where. Okay. So initiation, then elongation story is controlled by something called super elongation complex. Once that pause has to be released, super elongation complex comes in and takes charge of it and does that. These two proteins I'll especially point out. These proteins, ELL, AF9, ENL, I show that proteins there, right? Leukemia, childhood leukemia that occurs because of translocation of those proteins. So there's a lot of work that goes on in studying these complexes in leukemia setting. Okay, so remember the story of my friend that I told you who died when she was 8 years. She was in the 8th grade. She died because of a TALL. Okay, the leukemia of T cells. That happens a lot, actually, by the way, in this part of the country, or as such in India. Okay, the childhood leukemia that we talk about. Like you guys have had kids dying out of cancer. That's leukemia. Okay, a lot of it. So this is really important, isn't it? So Mr. Debu works on identifying why do those translocations in AF9 ENL actually cause cancer. That's what he does. That's why Wellcome Trust gives him 15 crores of rupees to do the research. And he would have made like many houses with that, right? Right? But the lots and lots of money goes into research to be able to figure this out, because we care about patients, right? We care about biology. So this complex is critical to understand how this controls transcription elongation. It controls transcription elongation by recruiting something called PTFB. I just told you that. PTFB interacts with an oncogene called MYC. Have you guys heard of MYC oncogenes? How many of you have? Good. MYC is one of the biggest oncogenes in the field. MYC increases transcription of around 1/3 of all the 23,000 genes. So when MYC is amplified, means many copies of MYC are made, MYC actually talks to PTFB and will go on and increase transcription. So rather than making 10 copies of that mRNA, 7,000 or 8,000 of genes now will be making 10,000 copies. So think about what happened to the cell. It got loaded with mRNA that it didn't care about. That's called transcription addiction in cancer. Okay? So that transcription addiction needs to be resolved to cure cancer. Okay, cancers where MYC is amplified. What did I say? I said a lot of things. Did you guys, how much of that did you guys get? What does amplification mean? Anybody? What does amplification mean? I said translocation. Did anybody get that? No. What does translocation mean? Nope. Then I was just saying for no reason. What does amplification mean? Number of copies, that's right. So in cancers, for example, there is DNA, specific DNAs get amplified. MYC, wherever MYC code is coded for, that piece of the DNA gets amplified, meaning many copies of that now are made in cancer. That's called amplification. What does translocation mean? Translocation means change in position, that's right, but translocation will mean wherever the AF9 is coded from, for example, there is a next gene that comes next to it and sits down, and that together one will make a different protein, not a regular AF9, it will make a different protein. That's called translocation. Thank you. But those translocations occur a lot in cancers as well. Okay, so cancers' genomes are messed up. Cancer is a genetic disease, its genomes are messed up. So whatever genes I was showing you in IGV, that is not a cancer gene. That's a straightforward. Some of those regions are amplified. We can talk about that another time. So landmark discoveries in eukaryotic gene transcription. Who was asking about papers? You want to read papers? Go and read these papers. Okay. This is from last author. Where is the last author? Can you guys read the last author's name? Can you guys read it on there? No? It's a guy called Roger Kornberg. Okay. This is a crystal structure of RNA polymerase he solved. This is, I think, in 1997 or something like that. But sorry, that's not a perfect thing. So that's a crystal structure. They showed how this RNA polymerases work. How is the RNA polymerase 2 in pre-initiation complex, works, that I just showed you. Pre-initiation complex with all the TFQB, TBP, etc., etc. So these are different factors here, and how they work. It shows here actually on the bottom, right? You can see POL2, TF2E, 2F2A, F, all of that. These are different subunits, how they were assembled together, and how, where is the DNA? DNA is right here in between, how they sit on the DNA, and how they go on and make transcription initiation happen. Go read the paper if you want, okay? And then this is the RNA polymerase 2 structure. This is Roger Kornberg's paper. This is a Science paper. Science is the topmost journal in the field. And he got a Nobel Prize for this in 2007. That's what I was telling you, I'm making you clap for earlier. He also happens to be a PhD mentor for my PhD advisor. Okay, so my PhD advisor actually trained with him, he did his PhD with him. So Roger Kornberg solved the structure of RNA polymerase, and he made many important discoveries in this that we'll talk about. You should go read about him. Okay? Go read about Nobel Prize winners if you want. There is so many beautiful stories that you would hear. Okay? There was an Indian guy who won the Nobel Prize, right? Too? Huh? Govind Khurana was many years ago, predated your and my life. Okay, who else? Have you guys heard of Nobel Prize winners? How many Indian Nobel Prize winners? Indian names do not need to be Indian. Huh? What? Avijit Banerjee, the economist guy. I'm talking about biology, guys. Come on. I mean, all power to him. Sorry, that's not what I meant. But University of Cambridge, translation, Nobel Prize. What's the name of the guy? The South Indian guy. He's alive and kicking. He's in University of Cambridge. No? Sorry. See what happens with my brain. Venki Ramakrishnan. Have you guys heard of this guy called Venki Ramakrishnan? Shame on you guys. Venki Ramakrishnan. Please go read about him. Okay? He solved the structure of ribosomes. You guys know what is ribosome? He worked in Utah. I had the privilege of seeing him in Utah when I was doing my PhD. He was there. Please go look at that. Look at him. Okay, while we are at this topic, I want you guys to read about this. This is my colleague called James Ellison. Jim Ellison. He has a movie made after him in YouTube. Go and look at him. Why do I take this guy's name? He has solved something called immunotherapy for cancer that is actually curing cancer patients that we'd never heard of. Okay? You should look at him. He's a crazy guy too. These guys are all crazy. Okay? Somebody was asking me, right, what to read. Go read their papers. Go read these papers that I'm just talking about. So so Roger Kornberg came to Utah when I was there, and he was showing a picture of a guy being in these big reactors. You know reactors, biochemical reactors. To solve the structure that you just saw, an RNA polymerase, somebody had to use this whole big reactor to crystallize protein. And he showed a picture of a guy going upside down into the reactor. Okay? To make that happen. So, what you are seeing on the screen are amazing people who do amazing things, crazy things to get you what you just saw in a paper. A paper which essentially has changed the way we think about how gene transcription happens. Because it happens in every single gene, every single second. As I'm speaking, lots of genes are being transcribed in my body. As you're listening, lots of genes are being transcribed in your body. You are you because genes are being transcribed in your body. They are being translated by ribosomes. So Venki Ramakrishnan was a great guy who made an understanding of how the ribosome structure is. Okay, and you don't know him. But I'm sure you know the best Hollywood actor, right? Don't you? Like you guys think about who you appreciate in your life, who you respect enough, and what these people do. Of course, entertainment is very important. We all need to be sane. But you must appreciate people, at least know the names of people who probably are of your, of your nationality, if anything else, right? Anyways, sidewise. So, okay, initiation, pausing, and go. Did you guys get this? Right? This is just a summary of what I just showed you earlier. Basically, so PIC is the pre-initiation complex and assembly happens. TF2B phosphorylates. Then you have DSIF and NELF that allow the pausing to happen. And then lots of things come in and then, recruit this and go on, right? So that's when the elongation happens. That's transcription for you. I could play this movie for you if this will play. It's all, we're hitting 12 o'clock. But if in your free time you can go and make it. This movie is very important. Will give you really visual cues of how this whole thing actually happens. This is the work of many years by many people. This guy Patrick Kramer is the first author on that, which he got the Nobel Prize for. Roger Kornberg. First author means he was the one who did the work. Roger Kornberg was just sitting on the bench saying, go do that work, right? But Patrick Kramer is a great guy. Okay, I didn't play this. That was not the intent. I don't get money from Vicks VapoRub. This is okay. That's all right. Ah, seriously. Okay, that was just entertainment for you guys. No, this is not what I played. Okay. Yeah, this is what I wanted to play. I'm sorry. Okay. So this actually is RNA polymerase. There is a DNA. I think, this will talk all about how this whole thing happens. You can go play this in your own free time. Playback speed. We're going to just play it at speed of 2. This is real protein, by the way, right? What was last year Nobel Prize was on? Huh? AlphaFold. Thank you so much. Somebody knows. What is AlphaFold for? AlphaFold is an AI, artificial intelligence based program for defining these structures. So now AlphaFold can tell you all about protein structure that people took years to do. And that can tell you pretty good damn, close to what real stuff is. So that's why it got Nobel Prize. So the world is changing, guys. The winter is coming. How many of you get that phrase? Game of Thrones. Everybody knows that, right? Winter's coming. AI is coming. AI is calm, right? You will not need any of us after some time. Okay, cool. You guys can play this on your time. I just want you to appreciate that there is a whole process that is given on this particular video that essentially tells you exactly how RNA transcription happens within seconds, like we know all the time, right? So please go ahead and this, again, they'll send you the slides, and there's a link here. You can actually play this movie. It's actually not relevant to what we're going to teach. But I want you to appreciate how complex the transcription is. Okay, so the GTFs are, what is GTF? Can anybody guess? General transcription factors. Great, thank you. So people went on and tried this in a test tube and said, boom, this should work, right? If this is how the whole thing happens. Guess what happens? It does not work. Okay, what does this mean? This means that there is a lot more to the beautiful transcription I just told you as a very simple process. In fact, that's just 1% of the whole thing. In fact, 5% of the whole thing. There's a lot more regulation to transcription, something called regulatory elements that affect gene transcription. Okay, it's 12 o'clock. Are you guys already done with me? You guys want to go ahead for a little bit more, or should I stop here? No? Yes? Oh, half the class is empty anyways already. This was full a few minutes ago. Okay, that's why I say I'm a horrible teacher, because half the people leave in between. Okay, cool. They were here just because somebody sent them to fill the class. Right? Isn't that true? Okay. All right. What I want you to appreciate is that transcriptional regulation was not just that simple. It's much more than that. It does have actually many elements in the genome that control the transcription. So that's what we call regulatory elements. Okay? Regulatory elements help us understand that transcription of this gene called RNA here, the BTA whatever, is actually modular in nature. How many of you know how a car is made? Yeah. Why am I bringing car when I'm talking about transcription? Right. Every engineering thing. Thank you. Every instrument is modular in nature, right? What does modular mean? Meaning I have an engine, I have a steering, I have a chassis in the car, I have a body of the car, and that's how I make car, right? In the engine, I have a battery powered system, I have a fuel ignition system, I have multiple things. That's modules. Any machine is made of modules. The same way, our RNA transcription control is made of modules. Okay, I want you to appreciate that's very simple. We are modules. We are essentially modules. Our eyes do a simple job. Our gut tube does a different job. Our legs do a different job, right? So we are essentially modular too. Every damn thing in this world is actually modular. This fan is modular, right? Everything is modular. So what does that mean? Why am I even talking about modules? Because you need to appreciate that modules are needed to bring signals together. Okay? Why do I care about signals in gene transcription? Talks about different signals. What do car modules do? My steering wheel gets a signal from my hand to move in a specific direction, that gives signal to the wheels to move in a specific direction, right? If I give hand this way, it's going to go this way. If I give hand this way, it's going to go this way, right? So my modules are controllable by my hand. In this particular case, my steering is controlled by my hand. So what am I talking about? Somebody stand up and tell me the answer. Why am I talking about modules? Why will cell make something modular? Oops, they didn't like it. Okay, what do you guys think? That means I was not being effective. Come on, guys. Why do I care about modules? In case of gene transcription, why do I care about modules? Good. Signals आते हैं. Good job. Signals आते हैं. Signal कहाँ से आते हैं? हाँ? Okay. Somebody else. Signal कहाँ से आते हैं? Okay, they come from adjacent cells. Yesterday night I ended with a slide. And metabolism, okay. What else? Environmental signals, right? The signal to the Chui Mui plant was my touch, right? So every environmental signal needs to control the gene expression. How does that signal get integrated to control gene expression? Through these beautiful modules that cells have built, right? Okay, did you get it? So this is a simple signal transduction electrical engineering phenomena, right? Modules, signal from here, signal from here, signal from here. I'm going to take the signal, I'm going to convert this to a signal that will be given to Mr. Kinase to do the phosphorylation. Otherwise it will say no more, I'm not going to go anymore because my kinase is not active. Where does my kinase get the signal from? These modules, they will give you the signal to say, okay, go on, phosphorylate RNA polymerase 2 or not. Is that clear? Okay, so what are the modules? The modules are so basic transcriptional status, apparatus, which we talked about. TBP, RNA polymerase, pre-initiation complex, Proximal cis regulatory module, which we call promoter. Distal cis regulatory module, which we call enhancer, right? We started the class with promoter and enhancers. Okay, is this simple, straightforward? No? So this is a schematic, a simple cartoon diagram, if you will, of things where you get signals from. Enhancer, what is enhancer? What does enhancer mean in English? I can't hear. One person stand up and tell me what does enhancer mean. Enhancer enhances, of course, right? Silencer, what does silencer means? Not the Munna Bhai MBBS silencer, right? The silencer silences, right? Okay. And then there is a promoter that helps give signal. Why are we putting pluses and minuses? Because those things give you signal to either go on, activate the gene transcription, or minus, suppress the signal from a silencer, or whatever, right? Something. Modules take the proteins, proteins give signal, signal starts the transcription, right? Simple stuff, right? Cool. I don't want you to think about what is TF2B, 2AH, 2E. That's how we learn our things. But I don't want you to think about that. That's not important. It's important for you to understand the concept. There are multiple modules that will control the signal so the gene transcription can go on. Those modules, why do I care about the modules? These modules are called epigenetic elements. Okay? All right? So silencer is a module which is an epigenetic element. Enhancer is a module that is an epigenetic element. Okay, promoter is a module that is an epigenetic element. Okay, all right. So we have epigenetic proteins, we have epigenetic elements built into the DNA. They are essentially modules to control gene expression. Okay, that's how an aldehyde dehydrogenase is made into liver. Neurotransmitter is made in the neurons. Is that clear? All right. We got some fundamental basics of things. Okay. All right. Here's a little more complex thing. This is also undergrad stuff, by the way. This is an undergrad book, right? I studied this in my undergraduate cell biology. I'm sure you guys did. Did you? No? How many of you really studied this in undergrad? Pura haan tha nahi, aadha nahi. Only a few of you have studied this in undergrad by cell biology? Okay, very interesting. So this is again a schematic from the book that I love, right? Molecular Biology of Cell or whatever. Tata box, we got this one, got this, gene regulatory proteins, got that, right? RNA polymerase 2, gene regulatory proteins. Gene regulatory proteins are epigenetic proteins that we'll talk about now in the next class, and the class tomorrow, and the class tomorrow, and the class tomorrow, right? Those are epigenetic elements. Those are the gene regulatory proteins. Something called this is really how the gene transcription works. You see how many proteins are there on the bottom? Can you see all these pictures of the red and the white and the thing and the thing and the thing? That's only very few of them actually. These complexes that control gene transcription every single time are many of them. This thing, big thing called mediator. Mediator, what does mediator mean? Who mediates the fight between the two things, right? Two people, mediator. Mediator mediates the signal from distal elements. When it's a dot means it can be 100 bases away, 5 kb away, 5 MB away in a different chromosome, can be 200 MB away. So gene regulation of a gene X is not controlled by the region next to it necessarily. It gets controlled by regions that stay far away from that gene in a physical location. That's why the looping is important. That's why chromatin structure is important, right? So why does 3D chromatin structure? We'll talk all about that in 2 days from today, but I want you to appreciate that this whole small little thing, right, gene initiation, transcription initiation, is a very complex process that gets controlled by many things. That's what I'm trying to get you. So the 3D chromatin structure is another epigenetic element. When we talked about yesterday, DNA methylation, histone modification, 3D chromatin structure, that's another kind of epigenetic element, okay? Actually, most of the DNA in our body is put in a specific conformation, and it's a controlled conformation. It's not random, okay? All right, that's a research that has happened in last 10 or so years. My lab does a lot of this as well. So anyways, we do it in the cancer context, but mediator makes RNA come, okay? Again, simplistically, I want you to get the concept. I'm not telling you what gene that is and all of that, right? And we can learn all about that and you can remember it, but today's world, you can Google things and you can find them out in 1 minute. This is a very old paper, and this is a summary from a very old paper. This is a 1989, okay? When many of you weren't born, maybe your parents were not born, right, by then. This is a gene called TTR, does something. I don't know what it does, to be honest. I think it's a liver gene. Basically, in this gene, why am I showing you this is very simplistically. Most genes actually have this kind of conformation where you have a gene, tada box. This is the promoter where specific transcription factors bind, HNF1, HNF4, HNF3, AP1, TBP beta. Okay, these are transcription factors that bind to specific DNA sequence. This is the real stuff. This is a gene called TTR that's actually in the liver because these HNFs are only expressed in the liver. This is some of those 2,200 transcription factors I talked about. Okay, this is that. So there are how many of them have to bind? 5 of them have to bind here. Okay, and then there is a dash here, which means there is more sequence in between, and then -1,8 to -9. So that's in base pair upstream of the gene. That's a non-coding DNA. That's called enhancer that we talked about, right? Enhancers mostly control gene expression. Okay, enhancers are the key to who you are, to why your I becomes I. In fact, people have mapped enhancers all over. Right, there is lots of effort, billions of dollars spent onto it. My own lab has spent millions of dollars identifying enhancers in cancers, but a lot of people have done it in regular tissues. Did you guys get this or no? Or am I just speaking because you're all hungry? You need food. Everybody needs rice and roti, right? No, noodles. That's another gene. Okay. Same way, just like what I just showed you, there is a different set of transcription factors on the bottom. This gene is expressed in the blood. I can tell you that. Okay. Beta globin. Beta globin is expressed in many places, by the way. I was wrong. That's a promoter. Okay. And there's an enhancer right here, which is about plus 2200 to plus 2400, meaning it can be downstream of the gene. I just showed you that to tell you that the enhancer can be anywhere, doesn't have to be upstream, can be downstream, happens. But bottom line is these enhancers essentially have binding sites for specific transcription factors. Okay, so your DNA sequence actually controls the gene expression. Okay, in cancers, these enhancers go at the wrong places. So that's how they control the gene expression. Okay? So my lab maps a lot of these enhancers. We have produced large amount of data in cancer space to identify where enhancers are, so that I can actually go on and build therapies against those beautiful enhancers that are unique to the cancers. Okay, all right. That's sort of a research which you do to understand what happens in cancer. So regulatory elements in eukaryotic genes influence gene expression. Why would regulatory elements control transcription? We just talked about it. To respond to the extracellular cues that could be of any kind. The food you eat, that you're going to eat in a little bit, right? That's going to control gene expression of things. That will make you sleepy. You eat a lot of rice and starch, that's going to make you sleepy, right? Will it? It will. And then you come back to my class and you won't get half of it, right? But nonetheless, that starch will control the gene expression of genes that will, hormones that will induce your sleep, right? That will happen in a daily life, guys. You're going to experience this in a few minutes, okay? Am I kidding? No, right? This is real stuff, guys. This happens all the time. You live, you are you because of gene transcription specificity, because you ate something that gets you something. You don't eat something, you're going to get hungry. You're going to produce cortisol, you stress, you're going to be angry, hangry, right? Right? That happens all the time. I'm not kidding, right? How many of you are angry at me right now that Kunal, let me go, man. I'm done with this shit, right? Enhancers are the ones that control that signal. Okay, so this slide on right now is telling you an enhancer element that is allowing the binding of these beautiful transcription factors, 3 little dots here, that will then recruit Mr. Mediator, which is a big complex that I showed you in that picture from MC. Did you get that right? The blue picture that was there? You right? Remember that mediator then will bring this enhancer close to the promoter and actually help increase the transcription, right? Okay, all right, let's go back to that. I want you to appreciate that this is what happens. Okay, so there is my enhancer in the green that beta transcription factor recruited a mediator complex that led to the gene expression. Okay, that's how it works. Everybody wants to go home? I think people are like leaving now because they're like, I'm done. Okay. This is my last slide. I'll let you go. So there are enhancers and something called super enhancers. Americans like everything in super, so they just call it super enhancer. This was discovered by somebody called Rick Yang at MIT. Okay, so they're called super enhancers because they have higher density of such transcription factor binding. This is called super enhancers. So why would that matter? Because in a typical enhancer, it controls the signal from a LIF, TGF beta, Wnt. Who knows these things are? What is LIF? What is TGF beta? What is Wnt? Cell signaling pathways. How do cell signaling pathways respond? They respond to the cues outside, right? Wnt is present, the Wnt is going to be activated, da da da. So cell signaling pathways, have you heard of them, right? Cell signaling pathways end up activating transcription factors. Boom! That's it. We got this now, right? You guys got this? How an extracellular cue eventually leads to gene transcription, specifically because it activates specific transcription factors. They can bind to specific DNA sequences, and that's how they will activate specific enhancers and thus specific genes. I will say it like specific like 5 times. Did you guys get that though? That's the key message. That's all I want you to get today. I want you to understand there's an extracellular cue, it gets a cell signaling pathway, the end of cell signaling pathway with these kinases that I told you about, a MAP kinase, the Wnt kinase, the whatever, that gets down to a transcription factor, and the end of those all signaling pathways is a transcription factor. Okay? Because they can't work if they don't control the gene transcription. And they control gene transcription through enhancers. That's why the specificity is that I told you about earlier. Everybody sleepy? Okay, all right, good night. That's that. This is just a summary slide. I think you guys got this, right? Okay, thank you. This is what you need to remember. But okay, so what did we learn? I just want you to summarize. We learned transcription factors, etc., RNA polymerase, etc., that get signal from regulatory elements like enhancers, silencers, etc. That's how epigenetics works. So we're done with the course. Okay? All right. Have fun. We'll see you in an hour or so.